and directly introduce these aniline amino acids just on the genetic level, and we can put them in each of the capsid protein monomers. And so now when this thing comes together, we have 180 of these pink spots on the outside. These are now our anilines that can participate in the coupling chemistry. Okay, sorry about that. So looking at how this works, we have a lot of examples now, but this is just one of them. So this structure here, this structure is a cyclic peptide. The specific kind, it's called an arginine glycine aspartic acid, or RGD peptide. What that means is that RGD is a sequence that commonly binds to proteins on the outside of cancer cells. It's one of the more commonly used ways that people target cancer cells. And so what we find is that if we have our aminophenol and we have our anilines and our capsids, in just 2 minutes we can couple this to almost all of the positions on the outside surface. We can read this out by these are the ones that haven't been modified and these are the ones that have, showing almost complete coverage on each of these spheres. And now we can test them to see if they bind to those cancer cells that bind RGD. We can also do this. These gels show that we can attach polymers. In just a few minutes, we can coat the entire outside with polymers that might shield them from being recognized by the immune system and so on. So we have a lot of opportunities now with this external coupling. So I'm just going to tell one story about this to kind of show what we can get by putting these two groups together. And that's shown here. And this is the work of Nick Stephanopoulos primarily, also with Gary Tong and Sunny Shao. And what Nick did is that he put on the outside of his capsids these DNA molecules here. These are DNA molecules. We put our aminophenol. We use the chemistry that I just showed you. We found that we could put over 100 of these on the outside if we want to. Nick usually puts about 20 or so copies on the outside, spaced out kind of like this. Now, this DNA molecule that he chose is a special kind. It's called an aptamer. And an aptamer is a sequence of DNA that folds up in 3 dimensions in such a way that it binds to a specific protein. And there are many of these now.